Welcome to the Bio and Beyond podcast. I'm your host, Satwik Nath, and today we're going to chat with Dr. Michael Higley from the Yale School of Medicine. He has studied behavioral neuroscience at Cornell and then completed his MD and PhD in the MSTP program in the laboratory of Diego Contreras at the University of Pennsylvania. He has also received many honors in his research, including a Sloan Research Fellowship, a Klingenstein Fellowship, and most recently, the NIH Director's Pioneer Award. Additionally, he has a secondary appointment in the Department of Biomedical Engineering and is a member of the Wu Tsai Institute. Without further ado, let's dive right in. So, why don't you begin by telling us a little bit about yourself?
Cool. That was a great introduction. I appreciate that. I will add a little bit more depth. I was interested in medicine and psychiatry as a high school student and did my undergraduate work at Cornell studying behavioral neuroscience there and really got into laboratory research as an undergraduate and spent quite a bit of time in the lab and decided then to try to do an MD PhD, which is a dual degree program that is pretty common across medical schools and is a roughly eight-year program where you do both medical and PhD training. And did that, but about midway through decided that I was less interested in medicine and clinical work than I was in lab work. And so the traditional career path from there is to do a residency and go on and be albeit research focused physician. Instead, I did a postdoc, which is the sort of more traditional avenue for those that just complete a straight PhD program. So I did a postdoc and then wound up taking my own position in my own lab here at Yale.
Well, that's really interesting. So, was there anything that was there a specific experience or anything that really got you interested in what you're doing now?
Yeah, I think, oddly enough, when I think as I said, I started out with an interest in psychiatry and um, I think the thing even from a very early age that struck me as sort of amazing is that our experiences, our appreciation for the universe, our ability to have these conversations and have ideas and create art and all of this, right? I mean, all of this comes from this like couple of pounds of lumpy gray stuff inside our heads. And so something about this translation from something physical to the sort of ineffable experience of being was really appealing to me. And I started out being very interested in more complex behavioral phenomena, consciousness, for example, and a little bit eschewed more basic studies of sensory processing and perception. These seemed maybe too simple for me. And what I uh, learned sort of early on in graduate school was, in fact, perhaps at the moment, these very complicated ideas are honestly a bit beyond our reach for understanding at a mechanistic level. And in fact, there's an enormous um, richness of information yet to be discovered, even in what might be considered fairly basic perceptual questions. And so I started to realize that we could start getting a handle on making real connections between how sensory information comes into the nervous system, how cells create representations that are ultimately useful for behavior. And so it's kind of grown from there.
Yeah, that's really interesting. And yeah, I see how that could really interest someone in a field like that. I mean, just thinking about that makes me interested in that too. Uh, so you did work on inhibitory GABAergic circuits.
Yep.
Would you like to tell us a little bit about that? It's called GABAergic, right? Or is it GAB?
Yeah. Um, so Right. It's probably helpful to have a certain amount of background. Right. So my lab is largely focused on the neocortex, which is the part on top, which is most developed in humans. In fact, it's so developed that's probably what makes us interesting, at least relative to other mammals. Certainly allows us to have these kinds of conversations and do things like build rocket ships and governments and such. Um, and so the neocortex has a lot of different kinds of cells in it, neurons. A neuron is a brain cell. The substantial majority are excitatory, meaning they communicate with other neurons and sort of make those downstream neurons more likely to be active. They use a molecule called glutamate as their main neurotransmitter. But about 20% of the neurons in the cortex are inhibitory, and so their activity makes their downstream targets less likely to be active. And they use a transmitter called GABA, and so they're called GABAergic as an adjective. What's a little bit interesting chemically is that glutamate and GABA, which are the sort of opposing forces, opposing transmitters in the cortex, sort of the yin and yang of cortical function, are almost identical molecularly. They're both amino acids or amino acid derivatives, and so if you know your organic chemistry, the difference between GABA and glutamate is just one carboxylic acid group. And so there's an enzyme that just pulls that carboxylic acid group off glutamate, and you've got GABA. And so it's remarkable that you get these opposing forces that are mediated by very, very similar molecules. And so maybe because of their prevalence, most of the history of neuroscience, or at least neuroscience that studies the way synapses, which are the chemical connections between neurons, studies the way these synapses work, has been focused on glutamatergic signaling. We've uh, overall known less about the function of GABAergic inhibitory synapses, and so seeing maybe a little bit of a gap or something to be explored there. When my lab started in 2010, we decided to spend a bit of focus on these GABAergic interneurons and their inhibitory synapses that they form. So to do that, we take a lot of different approaches. Um, some of what we do is in called ex vivo, or outside of the living animal. We prepare brain slices from rodents, and you basically use a fancy deli slicer to cut very thin sections of tissue, and those cells can stay alive for several hours, and allows you to record from them and get a very detailed understanding of their activity and their mechanisms of function. And so we've done that to characterize various properties of these inhibitory GABAergic synapses, to characterize a little bit of the diversity. So it turns out there are a whole lot of different kinds of interneurons, and they have slightly different functions that we've tried to unravel. And then we've also done work in vivo, where we use electrophysiology and various types of microscopy techniques to image neural activity, often during behavior, and again understand how these different kinds of cells, glutamatergic, GABAergic interneurons, interact to support behavior.
That's interesting. So, what would you say are some of the things you've learned from these glutamatergic and GABAergic interactions from your research?
Yeah. One of the things that we learned early on, that I think still continues to percolate a little bit in my own lab, and maybe others as well now, is for a long time, GABAergic inhibition was viewed as a little bit of an unselective brake on activity. Like the purpose of inhibition was to stop other neurons from being active, to stop them from firing, we would say. But when you dig a bit deeper and you start to appreciate the diversity of interneurons, there are some types that make really powerful synapses on their target cell bodies. And again, a little bit of background. So, so neurons have very complicated structures. They have a cell body, which is kind of the heart and soul of the cell. But then they have lots of dendritic processes where they collect inputs, and those inputs can be located very far away from the cell body. And so there's a lot of integration of thousands and thousands of inputs that come in, and it's all integrated at the cell body, and then the cell body kind of decides if the cell is going to fire an action potential, and if it does, then that travels out the axon to communicate with other cells. So for a long time, what was mostly appreciated was that GABAergic inputs to the cell body were really well positioned to just turn the cell off or say, okay, you're less active. That's it. But then we find that there are many kinds of interneurons which, in fact, make their connections way out in these distal dendrites, kind of where all of the excitatory inputs are coming. And one of the things that we discovered was that those inputs, rather than just shutting the cell off, seem to be more important for fine tuning which inputs are important, at least for a particular moment in time. I would say they seem to shape the integration of all of those inputs even before they reach the cell body. And so again, a little bit that inhibition is a little bit less about just shutting the target cell off and more about sculpting the way the target cell integrates all of its inputs. So that subtlety was something that I think wasn't fully appreciated, and some of our work helped make that more clear.
Yeah, that's really interesting because when I was learning about this in psychology, I was learning more about how it basically slowed everything down. I didn't really learn much about this sculpting, how everything actually works.
Yeah, it's you know, just some really interesting back of the envelope numbers are maybe fun to think about, right? Which is so a neuron in the cortex gets, roughly 10,000 synaptic inputs. So 10,000 roughly independent signals arriving. And each of the cells, which are mostly its neighbors, um, those are active roughly about once per second on average. So every cortical neuron is getting roughly 10,000 inputs per second, and it's constantly integrating all of these inputs. So it's funny. I talked a little bit a moment ago about some of the work that we do in ex vivo acute brain slices. And because that tissue's been removed from the animal, it's not, the cells are alive, but it's been really removed from its more intact circuit. Those are very quiescent. They're very quiet preps. The cells are not that active. And one gets a certain perspective of a cell as a quiet pond in which you're throwing a pebble, and you might throw a pebble and I throw a pebble, and we look at how the ripples interact. You're like, oh, that's integration, which is true. I mean, there's the biophysics of the cell govern how those ripples, those inputs interact. But the truth is, when you look at that same cell in vivo in the intact animal having some behavior, it's not a quiet pond. It's like being in the ocean in a hurricane, right? It's these are these 10,000 inputs per second. It's not one and one. It's just oceans of activity. Um, and so again, all of those biophysical principles are at play, but it's a completely different regime of activity than one often appreciates. And so this is a little bit of a challenge in the field, is because the preparations where we have the most ability to investigate fine details of cellular function lack a lot of the relevant amounts of activity. But the preparations that have all of this sort of normal behaviorally relevant activity, it's methodologically much, much more challenging, sometimes bordering on impossible to um, to to get the kinds of fine-tuned experimental data that we might want. So you really have to try to triangulate on the truth by using different approaches.
Yeah, that's really interesting. And it also gives a new perspective because all these, when you take a slice, yeah, you only get a certain amount of neurons that are giving data, not all the neurons in the full cell, which would actually make it I guess it would make it a lot like throwing stones at a pond, like you said,
Yeah,
compared to a hurricane.
yeah.
Well, moving on from that, would you like to tell us a little bit about your research on neuromodulation?
Yeah, yeah, we can maybe make a nice transition there because there's maybe a semantic distinction between what we might call a neurotransmitter or neurotransmission and what we might call a neuromodulator or neuromodulation. I think the distinction is a bit subtle and perhaps not all that useful, but nevertheless, we think about transmitters like glutamate and GABA as being very clear, go, stop signals. I excite you, I inhibit you, and that's sort of straightforward. Neuromodulators, of which there are, many, many substances that are classed as that, probably some that much of your audience will at least have heard of. Dopamine, often simplified to being like the reward chemical. Serotonin, which is implicated in a lot of mood disorders, certainly many of the medications for depression and anxiety modulate or regulate serotonin signaling. Um, Oxytocin, acetylcholine. There's a number, and the reason they're called modulators is that they often don't directly excite or inhibit the target cell. They make the cell more or less receptive to those start and stop signals. So, if you sort of magically took all of the activity of the brain away, the modulators might not um, have any obvious impact. But in the context of these normal excitatory and inhibitory signals, modulators can subtly fine tune the way a cell responds to its inputs. They can make those inputs a little stronger, a little weaker. They can make the cell a bit more or less excitable, meaning that, for the same impulse, it has a bigger response or a smaller response. And so for that reason, they're called modulators. There's, far more that we can talk about at a biochemical level, but that's probably a broad distinction. And so some of our work um, has been in vivo, in this case, in looking at intact behaving organisms. And one of the things that we and many other labs have found is that even in an animal that's not obviously doing something, it's not trying to get a reward, it's not trying to escape something, it's just kind of chilling and hanging out. Um, there are a lot of transitions in that animal's behavioral state. It can go from a level of high arousal where maybe it's really kind of paying attention to what's going on around it. It's really focused on something new to moments when we might say it's more quiescent, that it's relaxed, maybe it's a little bit less attentive. I sometimes think about this a little bit on watching sports on the weekend, is that sometimes you could be sitting on the couch and you're not asleep, but you're not exactly the most attentive that you might be. You can imagine another moment sitting on the couch, somebody comes into the front door and suddenly you're really focused on like who is it, and so it turns out, these behavioral states um, map onto different patterns of activity in the brain, especially in the cortex. And these different patterns of activity and the consequences for one's ability. Again, when you're alert and awake and somebody asks you a question from the other room, you hear them, you respond. You're kind of dozing on the couch in the middle of a game, and somebody asks you a question, and maybe you don't hear them, or it- those differences, those state-dependent differences, are driven enormously by changes in neuromodulation and things like acetylcholine and dopamine and norepinephrine. And so some of our work we've really done a couple of different kinds of studies. We've monitored neural activity either through electrodes or various forms of microscopy. And we've used pharmacological agents, drugs, to to manipulate, say, acetylcholine, cholinergic signaling, and ask what are the consequences for neural activity. And then more recently, and maybe even more excitingly, um, different labs throughout the world have developed reporters that light up when one of these modulators is released. And so we published a paper a couple of years ago now looking not only at how does acetylcholine change neural activity, but how does behavior change when and where in the brain acetylcholine is released? Because we have these reporters that light up in the exact location where acetylcholine is released. And so you can relate not only the function of these modulators to neural activity and behavior, but you can really understand the dynamics of the modulators themselves.
So basically, you would be able to understand what is, you would have a much deeper understanding of what is going on at that time using this new technology.
Yes, with extremely high spatial and temporal resolution. I just put numbers on that. Spatial resolution of signals within, a few microns or a few tens of microns of each other, a micron being a millionth of a meter, um, and spatially within a few tens of milliseconds.
That's really interesting. That would mean it's extremely accurate.
Yes, although it's funny, I mean, accurate, but sometimes even now, some of the signals are measured um, still indirectly. So we measure the dynamics of signals and we have the resolution that I mentioned, but a lot of times, take for example, some of the fluorescent indicators that we use, they're relatively slow. So when a molecule, say of acetylcholine, binds the sensor, the signal that we see might take a little while, meaning a few extra tens of milliseconds, if not hundreds of milliseconds, to evolve. And so the thing that you measure is in fact a slightly slowed version of the underlying truth. And there are mathematical ways to try to recapture the underlying dynamics of the actual signal. So on the one hand, we have extremely high spatial and temporal resolution, but on the other, there's still often indirect readouts of the underlying biology.
But you said it's still like tens and hundreds of milliseconds. That's still relatively fast, no?
It is, although your individual brain cells can probably make distinctions on the order of a few milliseconds. So
Mm. Well, that's really interesting. And where would you see this research being used? Like, in what ways could it be applied in medicine and technology?
yeah, I think that's an important question, maybe especially the present moment in history. I mean, the truth is that most of our greatest engineering feats and scientific discoveries, understanding about the universe, some of which simply gratifies our sense of being alive, but some of which has gone on to to shape our ability to treat and cure disease, came from extremely basic scientific discoveries where the initial scientists did not have any idea that what they were working on might have transformative benefit, and that wasn't really their intent. So, on the one hand, it can be it can be difficult to ascribe clear um, future benefit from any one line of basic science research. But on the other hand, history tells us that most of the most important discoveries started out that way. And so there's a danger in demanding that everything have very obvious benefits right now. That being said, one of the challenges, maybe this is something that drives me a little bit. My lab works to some extent on both neurodevelopmental disorders like autism and schizophrenia and on neurodegenerative diseases like Parkinson's disease. Much of the disease modeling and our understanding of disease comes at the molecular and genetic level. We begin to understand which genes are associated with an individual having a particular condition, or why cells in neurodegeneration, neurodegenerative disorders might might suffer and ultimately die. We really don't have a great understanding of why some of those molecular changes should lead to the particular phenotypes that they do. Um, as an example, right? Schizophrenia and autism, from a clinical perspective, look absolutely nothing alike. I mean, they're completely distinct disorders, but they both share, in a general way, dysfunction of synaptic connections in the brain and probably in the cortex. So that's what we don't really understand is it's one thing to go to the textbook and say, oh, both autism and schizophrenia are synaptic disorders, but why do they look completely different? That we don't understand. And so the hope is that, some of our experiments where we're trying to ascribe particular functions of particular cells and synapses to particular behavioral outcomes will allow us to start to untangle why disruption of particular genes, particular molecular pathways give rise to particular, behavioral disorders, and eventually, give us insights into how we might, ameliorate the challenges facing those patients.
That's really interesting. And yeah, I agree that from what you were saying, I would say that's probably one of the biggest things that this could be used for. But on the flip side, how would you say that advancing technology can help this research?
Well, a lot of what my lab does is based on advances in technology. I suspect it's technology without an enormously broad audience because it's sort of within the scope of neuroscience. But as I think I mentioned a couple of times, my lab uses microscopy
Hmm.
extensively, and for us that means finding on the one hand new molecular fluorescent reporters that label particular kinds of cells or particular kinds of activity. And so people are constantly, especially, molecular biologists are constantly developing new tools for reporters. On the other hand, we're always in search of better microscopes, better optical tools that give us higher resolution, allow us to see deeper into the brain. And so there's a lot of technological development on that side as well. One thing that's maybe an interesting point is so when one uses a microscope, right? I mean, there are lenses involved. There's a hardware to directing light through the microscope from your sample into your eyes, your camera, whatever you're using, and those physical elements, lenses, for example, can also be the tissue that you're imaging into, cause what are called aberrations, meaning they disrupt the way the light moves in a way that degrades your image quality. This is exactly the same problem that astronomers face when one is trying to see stars, where, on the one hand, space is empty, but over the distances that one is trying to see, there's actually a lot of stuff between us and like the star you're trying to look at. And so astronomers have developed an enormous number of tools, one category of which is called adaptive optics, where instead of just using a lens, which is kind of uniform throughout, you can in fact change the way light propagates through your optics to correct for aberrations induced by various elements along your imaging path. So this technology of adaptive optics has percolated into biology and now allows us with our microscopes to see incredibly deeper into the brain with incredibly improved spatial resolution than we might have had before. And so this kind of technological cross-pollination between disciplines is I think it's extremely important and uh, I think could certainly always happen more. The advent of AI, which in a way is a little bit too soon to say exactly how it's going to impact scientific research. I think it's a given that it will. It's more that the details of exactly how it turns out to be best implemented to be most useful, still have something to be worked out. But it has enormous power, I think, to collate and distill a lot of information quickly. There's a certain trust but verify when you're dealing with the output of say ChatGPT or some of the newer chatbots. But in the sense that it can give you a very good first place to start, and then you go and follow up and make sure that what it told you is sort of accurate. I think that's incredibly powerful. And so, often what we do is to try to comb the literature to make some quick hypotheses that we can design experiments around. Having some support in digesting large amounts of data quickly and helping to formulate um, clear, succinct hypotheses is likely to be something where, new developments in artificial intelligence will be quite helpful.
Hmm. And I agree. Artificial intelligence, although now it's still in like its basic forms, I believe that in the future, yeah, we probably could be able to use it to help advance these fields, possibly help us find out more about these, well, in your work, like all these inhibitors like GABA. And although we're probably nowhere near that yet, I believe that in the future we would be able to do this. Um, and last question before we end this. Do you have anything that you would like to say to the audience watching this who are interested in neurology and perhaps want to move on in the future and work on neuroscience?
Yeah, absolutely. I mean, the scientific enterprise is both enormous and diverse and has an incredible number of different ways that you can get involved. From medicine, becoming a physician, becoming a nurse, becoming an allied health community member, to research, doing the kinds of basic science that I do, running clinical trials to understand the efficacy of various drugs or other treatments, to teaching, to communicating science to a broader audience. We need more and more of this, and it is shocking for many of us in science to feel like the scientific discipline itself has lost a certain amount of respectability or support these days in society. And the reasons for that, I think, are complicated. But uh, maybe the thing to say or to end on is that virtually all of us in science, and there are always some bad apples, I suppose, but the vast 99.9% majority of us are really just interested in figuring stuff out. um, we certainly, one does not go into science for the money. One goes into it because you are just curious about how things work, and you like discovering things. And so we get lots of things wrong, but we get lots of things right. And this notion of scientific discovery is really an evolution of how we understand the universe. And so when we get things wrong, we change our models, we keep going. And so I think it's a noble endeavor in that regard, that we're trying our best, and we're trying to figure out, our universe and our lives. So I certainly encourage anybody that's interested to get involved in science in any way that they can.
I agree. And as we wrap up this insightful episode of the Bio and Beyond podcast, I want to extend our sincere gratitude to Dr. Higley for sharing his wealth of knowledge and expertise. His commitment to the field of neuroscience is truly inspiring. Stay tuned for more captivating conversations at the intersection of biology, medicine, and technology. Until next time, this is Satwik Nath signing off from the Bio and Beyond podcast.
Thank you.
Remember, the exploration never ends. There's always more to discover.